Wird aber jetzt übertragen und das ist wahrscheinlich verzögert. Also ich muss Play drücken wahrscheinlich. Und dann Livestreams through. Das ist jetzt aber der Partagé. Und jetzt muss ich auf Zoom und ich muss Partagé l'écran, Share Screen. Share Screen. This one. Share. Et maintenant? Alors.
Jetzt, man sieht es jetzt.
Es muss auch noch hier sein. Es muss hier kommen vor allem. Oh, jetzt habe ich das hier.
Es geht ein bisschen gesprintet.
Okay, ich will das haben.
Jetzt.
Okay, gut. Das mache ich so. Wo bin ich jetzt? Ich will hier in Controls. Und das will in mein Fenster. Aber jetzt share ich was? Das? Nein. Sharing Screen. Hier in dieses Fenster gehen. Okay, mache ich Stop Sharing halt. Stop Sharing halt. Ich muss nochmal share. Also ich will diesen sharen. Share Screen. Das will ich.
Ist da nicht ein Präsentationmodus Share?
Warte kurz. Ich brauche das. Und das ist, was man verzögert sieht. Ich brauche ein bisschen Konzentration kurz und dann. Okay, das muss hier kommen. Gut. Und das.
Natalie is already there, I see. Okay.
Somebody's there.
Yes.
But
you
have any trouble?
No, not really, but I would like that I have to go back into my meeting, Zoom meeting, but
you don't know where is your Zoom meeting?
Here.
Yes.
But I don't. I want to go. Why do I have this?
This is your Zoom meeting.
But I want to. This is not my Zoom meeting because this is
yes.
This one.
Yes. You're sharing screen. What do you want to see?
Ich will die Sicht haben, dass ich klicken kann. See present a view. Aber.
See present, and there's no one of the options is not here. Here, new share. These are the settings of Zoom.
New share. Das bin ich am Sharen, oder? Share, nein. Das will ich sharen. Aber wo ist mein...
So if you stop sharing, you probably get this.
Das.
But when you are sharing, you don't see this, you see the little boxes here, yes. And then you see the panel, which is hidden.
Exactly.
Okay, good. So I wait with share my screen, so what we see on YouTube is not this.
Yeah.
But okay.
On YouTube, okay.
YouTube, sure. So you can ask somebody what they are seeing.
This days, that's the YouTube what you see actually. Yeah. Okay, but it's not a problem. So I can start sharing my screen.
Okay. So now Eric Tabaki is here. Okay, so Zoom is already, sorry,
Zoom is-
YouTube was already open.
It's already open. YouTube and Zoom is open.
The microphone is
Muted.
muted?
Yes.
Your microphone? Okay.
Should
Okay,
I,
Natalie, and maybe could you stop the YouTube a bit just to have a few words with
No.
you cannot.
Because if I stop it, it will be away. Like it's
Okay.
yeah. So
Okay, so maybe I can test. Oh, ok, donc on a nos deux membres de jury qui sont là.
Salut Nathalie.
Bonjour.
ah oui, il s'attend.
Ah, oui.
Oui, il s'attend.
Mais ce qu'il y a, c'est que le YouTube aussi, tu vois, il faut un peu de prix, tu vois, il faudrait un peu.
Oui, mais c'est là maintenant le problème que j'ai déjà commencé le YouTube pour être prête. Et maintenant, alors.
Donc, elle a lancé un YouTube.
Mais c'est encore,
D'accord.
ce sont 5 minutes, alors nous avons peut-être.
Donc, on a nos deux membres du jury qui sont là, mais voilà, c'est très bien qu'ils vont nous entendre. Je pense qu'ils nous entendent. Tu entends bien, ouais.
On vous entend.
Salut Eric. Très bien.
Bonjour.
Très bien. Ok, donc sans doute, bonjour, je suis Anne Charpentier, pardon, je me suis pas présentée. Et donc voilà, malheureusement pour la configuration de cette visioconférence, vous allez pouvoir voir Kathrin qui va être sur la... Par contre, les autres membres du jury, on va être, vous n'allez pas nous voir parce qu'on va être dans le public pour pouvoir voir l'écran de présentation. Je pense que c'est ça le plus simple.
Mais en fait, oui, peut-être nous pourrions faire un test si vous êtes là, si nous écouterons.
Non, mais moi, Eric, je ne veux pas le voir parce que ça lâche des jeux de bonjour comme ça.
Moi, c'est pareil.
Bonjour Nathalie, bonjour Eric.
Salut.
Ouais, l'important, c'est qu'on vous entende.
Ouais. Donc, donc peut-être pour le début de la présentation, avant que Kathrin commence, on pourra être là. On se met là pour introduire tout le monde. Okay. Nous, ensuite, on va se mettre au premier rang. Ah, okay. Et après, pour la discussion, on pourra avoir un micro.
Oui, mais le technicien, il m'a dit que en fait, le micro pour la visioconférence, c'est ça et pas ça. Hein. Mais il m'a dit ça il y a 15 minutes. Alors,
peut-être si on peut tester, oui, peut-être on peut s'asseoir là-bas pour voir si quand on parle.
Vous montez la voix loin. Vous nous entendez si on parle comme ça?
Si on parle moins fort, vous m'entendez toujours?
Oui.
Ouais,
Nathalie,
ça va.
Ça va?
Ouais.
Ça va.
Si jamais on ne vous entend pas, on vous fera la remarque.
D'accord, ok, désolé, ce n'est pas une configuration très, très agréable pour vous parce que vous ne nous voyez plus.
On imagine que pour vous non plus, c'est pas très pratique, mais bon, on va y arriver.
Ok. On reste dans cette configuration alors.
Pas de problème. Alors, j'ai parlé avec le technicien et il m'a dit qu'il n'y a pas une autre possibilité. C'est ça, et normalement, ça va. Mais s'il y a des questions pour la salle, nous pourrions faire...
Il n'y a pas de questions pour la salle.
Et je pense...
Non, il n'y a pas de questions pour la salle.
Alors, c'est ça, et...
Ouais, l'autre option, ça serait pour la discussion, mais il faut savoir qu'il faut qu'on bouge.
Manol, vielleicht du kannst den Leuten sagen, sie können reinkommen.
Pour la discussion.
Du kannst es öffnen. Ja.
On fait une petite pause et on monte sur la scène pour qu'ils puissent nous voir.
Ah oui, ce sera plus convivial.
Plus convivial. Ça vous va pour
Donc, à la fin de la présentation, on prend 5 minutes, là, on monte avec...
en fait, ah non, c'est pour la vision. Je pensais pour mute, we can mute it here, but then it's also muted for the Zoom.
Ouais, for the Zoom.
We mute it for the whole community or just.
Okay. It's okay, but we don't need to mute it. It's okay, so fine. So you do your presentation like that with this configuration, and after your presentation for the discussion, we will try to come with you on the stage to have for Natalie and Eric to have the possibility to see everybody. It's going to be much more nice. Okay, so now we let the people enter.
Yes, we let the people enter. Good.
Eric, j'ai une question à te poser.
Oui.
De quel institut tu dépends pour te présenter?
De quel institut?
Ouais.
En fait, je travaille à l'INE, CNRS INE, mais j'appartiens au laboratoire d'écologie fonctionnelle et environnement à Toulouse.
Ok. Merci.
Je t'en prie.
Bon. Alors, bonjour à tous. Nous sommes là pour écouter aujourd'hui Catherine, qui va présenter ses travaux de thèse. J'introduis en français, mais la suite va être en anglais, c'est-à-dire que la présentation sera en anglais, ainsi que les questions que les différents membres du jury que je vais présenter juste après vont poser à Catherine seront également en anglais. Donc, toute la discussion autour de la thèse se fera en anglais, avec notre piètre anglais. Tu nous excuseras par avance.
Oui.
Alors, dans l'ordre des questions, donc, après ta présentation, Catherine sera interrogée par ces deux rapporteurs qui sont en distanciel. Donc, vous avez le professeur Nathalie Machon du Muséum national d'histoire naturelle. Et puis, vous avez Éric Tabaki qui est chargé de recherche au CNRS, qui donc ont été chargés d'évaluer dans le détail ce travail de thèse. Ensuite, Grégory Mailly de l'Université de Liège poursuivra les questionnements en tant qu'examinateur, donc avec un point de vue plus distancié sur ces travaux-là. Et Anne Charpentier, qui a dirigé ces travaux, clora les questionnements. Et on m'a désigné comme président de ce jury, donc je poserai aussi quelques questions avant que nous allions délibérer un petit temps et qu'on revienne vous retrouver pour prononcer le verdict tant attendu. Voilà, donc pas de questions pendant, bien entendu, la séance qui va suivre. Pas trop de téléphone portable branché, s'il vous plaît, pour pas déranger Catherine. Et puis, tout devrait bien se passer.
Oui, merci.
Alors, je ne me suis pas présenté, moi je suis Franck Richard et je suis professeur de
Okay, do you see the screen? Nathalie Machon and Eric Tabaki. Do you see the screen? Good. Okay,
yes,
good.
perfectly.
Very good. Okay, good. So, yes, welcome to the presentation of my PhD thesis, in which I investigated the permeability of European protected areas to non-native species. First of all, it is a pleasure to have you here in person and online. This thesis was supervised by Anne Charpentier from the Centre Écologie Fonctionnelle Évolutive here in Montpellier, and co-supervised by Will Simonson from the UN Environment WCMC and by Kevin Smith from IUCN. I will give you a short introduction to the topic and then guide you through the 3 research chapters that I did, and end the presentation with discussing some limitations and perspectives of the thesis. Good. So we live in a globalized world where dispersed communities have been integrated in a worldwide community. This has brought some advantages, such as the transportation of goods and people around the world. Think of your holidays, of your computer that you ordered from China. But this has also led to translocation of species among ecosystems where they have not been present before. In these new ecosystems, these species are non-native species, and some of them establish, spread, or even can become invasive. This translocation has led to an accumulation of non-native species worldwide. And here you see a graph that shows the number of first records of established non-native vascular plant species per region. So what are non-native species? Non-native species are species that are introduced to areas beyond their natural distribution and biogeographical barriers. This introduction can be intentional or unintentional, but is always associated with human activities. For successful establishment in the new environment, these species need biotic and abiotic conditions to establish, such as, for example, the availability of resources or the availability of niches that to establish. But they have also the advantage that they have escaped their natural enemies from their native range. So not all non-native species have a negative impact in the new environment, and only a small subset of these species has the potential to become invasive. So what is the difference? Invasive non-native species are non-native species that have a harmful effect on the economy, environment, or health. Looking at environmental impacts, for example, they can compete with other species for resources, for niches, or they can even predate them. And thus, non-invasive species are among reasons for global biodiversity loss. In my thesis, I focus on Europe as a study region, and Europe holds a list of invasive species of unique concern that holds at the moment 66 species. To protect natural habitats and areas and ecosystems from threats such as from human degradation or habitat loss or non-native species with a negative impact on the environment, protected areas have been established all around the world. Protected areas are clearly defined biogeographical spaces, recognized, dedicated, and managed to maintain a long-term conservation of nature with associated ecosystem services and cultural values. Protected areas are often remote from intense human activities, and thus they are a refuge for native species, threatened species, rare species, or endemic species. Worldwide, more than 15% of terrestrial Earth surface are protected protected areas already. And even more, Europe is one of the regions with the highest number of protected areas worldwide. To look at protected area, one has to consider that protected areas are not isolated systems. They are systems that are connected with their environment. For example, through a directionality of fluxes, such as you see here, for example, by rivers, watersheds, or migratory corridors that connect protected areas with other habitats, or here shown as black dotted lines also by nearby communities, etc., roads, etc. So we learned that protected areas are not isolated systems, but however, little is known about the non-native species or environmental conditions in the surrounding of protected areas and how these affect non-native species in protected areas. Thus, the main question I wanted to answer in this thesis was, how effective are protected areas at resisting non-native species colonizations? Why is this important? It is important because protected areas aim to protect pristine natural habitats, and what you don't want to have inside these areas are non-native species, and most of all non-native species with a negative impact on the environment. However, looking at protected areas itself, it doesn't tell us so much why they are permeable to non-native species colonizations. It might be that they only show a part of the big picture of non-native species that are in the surrounding areas. Throughout the thesis, I investigated the presence of non-native species in protected area by investigating non-native species and environmental conditions in and around protected areas and how these affect the presence of non-native species in the protected areas. The general objective of this thesis was thus to understand factors that influence the presence of non-native species in protected areas. I looked at 3 factors, such as in my first chapter, at the pool of non-native species around protected areas. In the second chapter, on the land use around and inside protected areas. And in the third chapter, I looked at disturbance variables from around and inside protected areas, and there's the influence on the presence on a specific invasive species. The first chapter, the pool of non-native species around protected areas. Previous studies have addressed the questions of how effective protected areas are at, for example, resisting biological invasions. The non-native species presence was always linked to anthropogenic factors such as road densities or population densities in the protected areas or outside the protected areas. But however, there is a lack of knowledge about the non-native species pool that is in close proximity to protected areas. And thus the objective of this chapter was to investigate the pool of non-native species around protected areas and how this influenced the community of non-native species in the protected areas. I had 3 hypotheses. The first one was that non-native species in a protected area are a subset of non-native species around protected areas. The second one was that the number of non-native species in a protected area can be predicted by the number of non-native species around it. The third hypothesis was that non-native species are recorded earlier around protected areas than inside them. To do so, I investigated as a case study Norway. Why Norway? Because Norway has one of the most comprehensive databases on biodiversity data, including data from non-native species. This data is composed by or contributed by research institutes or environmental agencies, but also by citizen science data. And the advantage of these data of the non-native species was that they were impact classified based on their impact on the environment. And thus this was an advantage that I didn't want to miss in this analysis. I looked at terrestrial and freshwater species for animals and plants and records between 1950 and 2020. I had in total 1,602 non-native species in my database. I looked at protected areas from the World Database on protected areas, including protected areas larger or equal 1 kilometer square and only terrestrial protected areas. In total, I included 671 protected areas in this database. I mapped the belt around these protected areas from 0 to 5, 5 to 10, and 10 to 20 kilometers. So the first hypothesis, non-native species in protected areas are a subset of non-native species around protected areas, and thus they might have a qualitative similarity. To compare this, I compared proportions of taxonomic group, classes, and impact classification categories of non-native species in the protected areas and in the belts. And what we found first of all, most of the protected areas were actually empty. Around 80% of protected areas didn't have any non-native species present inside. But those who had, we found that in average, 63% of non-native species present in a protected area were also present in the associated 5-kilometer belt. This graph shows the proportion of non-native species by kingdom, plants and animals in protected areas and in the belts. And we found that more than 75% of the non-native species in protected areas and the belts were plants, and the minority were animals. Further, we looked at the most frequent non-native species in the protected areas and in the belts. And we found that among the most frequent 9 non-native species, 5 of them were also among the most frequent 9 in the 5-kilometer belt. Moreover, we found that 7 of the most frequent non-native species in the protected areas and also 7 of them in the belt were actually classified as invasive. Branda canadensis, which is the first one on the left side, was the most frequent non-native species in the protected areas. So this Branda canadensis is the Canadian goose. It was introduced to Norway for ornamental reasons and for hunting in the 19th century, and since then spread or colonized areas that are similar to its native range, and it's classified as invasive species because it can hybridize with native geese species. We found also qualitative dissimilarities between the protected areas and the belts. And here you see a graph that shows the proportion of non-native species by the ecological impact classification. What we found here was that 40% of the non-native species present in a protected area, but only 12% in the belt were classified as invasive. So this is actually bad news for conservation. We explain this, that invasive non-native species have a higher colonization potential than non-invasive non-native species because they have characteristics that permit fast colonization and spread. In addition, we found that invasive non-native species were 4 times more abundant in the belts than in the protected area. The second hypothesis was that the number of non-native species in a protected area can be predicted by the number of non-native species around, and thus they might have a quantitative influence on the number of non-native species in a protected area. I applied a zero-inflated negative binomial regression model because this takes into account that there were a lot of zero values in my database, meaning a lot of protected areas were actually empty. We looked at the number of non-native species in a protected area as a function of non-native species in the 5-kilometer belt. And in addition, we included also variables that are associated with non-native species present, such as the mean accessibility of the protected area, the mean human population density, or the land cover, and the surface area of the protected area. And what we found was actually that non-native species in the protected area were among significant explanatory variables for the non-native species richness in the protected area, and thus it confirmed our hypothesis. The third hypothesis was that non-native species are recorded earlier around protected areas than inside protected areas. And thus, species around must have a temporal influence on the species in the protected areas. Why did we assume this? Because introductions are and spread is usually associated with human activities. And this is more the case around the protected areas than inside. I compared the years of first records in the protected areas and in the belts by applying the Kruskal-Wallis test and Dunes test for pairwise comparison. And what we found was that in the protected areas, non-native species were actually recorded 4.5 years in average later than in the surrounding 5-kilometer belt. However, we found no significant difference between the first records in the first records between the belts surrounding the protected areas. We conclude that protected areas are under high pressure from non-native species in their surroundings, and we emphasize the role of non-native species colonizations from the surroundings in shaping the non-native species communities inside protected areas in a qualitative, quantitative, and temporary manner. And thus, we strongly emphasize to expand the focus of non-native species management in the protected areas beyond their boundaries. And this chapter I could publish with my colleagues in Frontiers in Ecology and Evolution in the year 2021. With this, I come to my second chapter, the land use around and inside protected areas. Our world is covered by diverse natural landscapes that provide habitat for species. However, landscapes have been changed and shaped by human activities, which has led to a loss of pristine natural habitats with the species and biodiversity in those. It is shown, or past studies showed, that the land use associated with human activities have actually more non-native species present. And also, past studies showed that the land use type can facilitate or hinder species movements. Protected areas might represent islands in highly human-modified landscapes, such as you see here on the bottom left, a protected area that is in the middle of agricultural areas and surrounded by villages. On the other hand, protected areas can also be part of natural landscapes with already high conservation values. As protected areas are connected systems with their surrounding, they are directly or indirectly influenced by the land use and management practice around them. And also, as I showed in my first chapter, by the non-native species surrounding them. The objective of this chapter was to investigate effects of land use in and around protected areas on the non-native species in the protected areas. I hypothesized that the high diversity of land use in protected areas results in a high number of non-native species in protected areas. And second, that protected areas with human-modified land use have a higher number of non-native species than protected areas with natural land use. And third, that similar land use around and in protected areas increase the number of non-native species in the protected areas. Here I investigated 4 countries from Europe. Why did I look at these countries? Because these countries also had comprehensive databases on non-native species data available on mostly national levels. I included terrestrial and freshwater animal species and plant species, and I looked at protected areas and their 5-kilometer belt surrounding them. I used the land use product from Copernicus Coring Land Cover at the level 1, and I grouped them into natural and human-modified land use types. The first hypothesis, a high diversity of land use in protected area results in a high number of non-native species in the protected area. We expected this because we assumed that the high diversity of land use types actually provides more suitable niches for non-native species than homogeneous landscapes. We applied the Shannon Diversity Index on land use types and applied the Pearson correlation between non-native species in a protected area. And here the result. We found a positive correlation between the diversity of land use in protected areas and the number of non-native species. And thus, this confirmed our hypothesis. The second hypothesis, protected areas with human-modified land use have a higher number of non-native species than protected areas with natural land use. We expected this because we assumed that there is a low colonization cost for non-native species in highly distributed areas, and on the other hand, a high colonization cost for species in natural and undisturbed areas where no human activities have facilitated their introduction or spread. We extracted the dominant land use types of each of the protected areas and the belts and applied a ANOVA and Turkish HST test. And here the result. You see here on the x-axis the dominant land use types in the protected areas and the mean number of non-native species in the protected areas. And what we found is that human-modified land use types in protected areas had significantly more non-native species than natural land use types, with the exception here, water bodies. And I come back to this in a moment. We applied the same analysis for the dominant land use types around protected areas on the mean number of non-native species in the protected areas. And also here, we found a similar pattern. We found that dominant land use type surrounding protected areas led to more number of non-native species in protected areas if it was human modified. On the other hand, more natural land use had significantly less non-native species in the protected areas. Again, with the exception of the water bodies. So what is with water bodies? So water bodies are aquatic habitats, and they have special characteristics since they have a fluid characteristic which has a lack of barriers and facilitate the spread. So if you imagine, for example, a lake which is partly protected and the other part not, there is actually just no barrier for species to move between these areas. And second, in the land cover type category, water bodies actually, this is not only natural water bodies, it could also include artificial water bodies, which are the reservoirs or channels, which are associated with more non-native species because they are more disturbed. And third, the control of introduced species in aquatic habitats is actually challenging. Why? Because you cannot just apply, for example, poison on a specific location. It would affect the whole ecosystem, the whole aquatic ecosystem. So with this, I come to the third hypothesis. Similar land use type in and around protected areas increase the number of non-native species in the protected areas. We assumed that similar land use in and around protected areas increase the habitat connectivity and thus the species movement between them. And also that the land use around protected areas can act as a filter to select species that are already adapted to these habitats. We looked if the dominant land use type in the protected area was the same or a different one to the dominant land use type in the surrounding belt. We applied a Wilcoxon-signed rank test to investigate this. Here the result, what we found was actually contrary to our hypothesis. We found that if the land use type surrounding a protected area was different to the land use type in the protected area, there were more number of non-native species in the protected area. We investigated the interaction between all of the land use types in the protected areas and the belt. And here, first of all, I show you an axis, which is the x-axis shows the dominant land use type in a protected area. and the y-axis shows the dominant land use type in the surrounding 5-kilometer belt. And here is the plot to this axis. How you have to read it, it's the darker square is, the more number of non-native species are present between the dominant land use types that are compared. And what we found is that regardless of the dominant land use type in the protected area, the dominant land use type surrounding protected area, if it was human modified, had increased the number of non-native species in a protected area. We explained this, that we think that the tropical availability around protected areas must have a more important role in protected area permeability than actually the land use continuity from the outside to the inside. To conclude this chapter, we conclude that non-native species in protected areas are strongly influenced by the type of land use around them, and that a diversity of land use types provides more niches and habitats for non-native species, and thus this has implications on the non-native species management in protected areas. And we think that the landscape context is actually of importance. With this, I come to my third chapter, in which I investigated disturbance around and inside protected areas. Why are you looking at disturbance? Disturbance is human or anthropogenic disturbance or natural disturbance are known to promote non-native species colonization and establishment. And furthermore, protected areas are supposed to prevent from disturbances, and thus we conclude that protected areas resist non-native species colonizations. In order to understand the effects of disturbance, I was looking for a species to conduct a long-term study where effects of disturbance might be or become visible and measurable. Acacia turned out to be a species that is suitable for this analysis because it is actually visible from space. It is visible on satellite imageries based on the specific footprint or specific spectral surface reflectance in the blooming period. This study I conducted in central Portugal, where the species is widely spread. And some words to acacia. Acacia was introduced in Portugal in the 19th century for ornamental reasons. As you can see, it has beautiful yellow flowers, and since then is spreading unpredictably. And its invasiveness is attributed to its high resproutability, its high seed capacity, its native exclusion by producing compounds that are actually that hinder the growth of other vegetation, and by its high environmental plasticity. The effects of disturbance by fire are, first of all, acacia is also known to adapt to burnt and resource-poor land and cleared land, and effects of fire, for example, can break the seed dormancy and thus promote the germination of the seeds. Also, fire removes the vegetation and clears the way for the species to grow. Vegetation clearance itself provides new niches and makes resources available and leads to less competition with other species. In this chapter, I hypothesized that protected areas experienced less deforestation and reduced fire risk compared to areas around them. And also, protected areas must have benefited from programs to control Acacia dealbata. And thus, we hypothesized that acacia invasions has been slowed in protected areas. Here I looked at 5 protected areas and their 10-kilometer belt surrounding them in a region that is in central Portugal. I used satellite imagery time series from 2001 to 2020 using Landsat 7 in the blooming period from January to April. In addition, I collected data on acacia for training and validation purposes. I did the fieldwork in Portugal in March 2020. However, there was the outbreak of COVID-19 and I had to stop the fieldwork. Thus, I complemented my database by investigating Google Street View, by investigating the area in Google Street View and on Google Earth Pro, and thus collect more data like that. I applied a machine learning algorithm. I used the maximum likelihood classification algorithm on the 3 categories: acacia, forest, and pasture. And I looked at disturbance variables from 2001 and 2009 to 2019 using Modi's burned area pixel product and Hansen tree cover loss. In addition, I did a questionnaire that I sent to the person in charge of the protected area management to investigate the present, past, and future management effort against Acacia dealbata in these protected areas. And here is the results. So this map shows the distribution of Acacia dealbata between 2001 and 2020. In the study area, in the protected area, and in the belt. So each of these squares actually represents 1 year, and you read it from the top left to the bottom right. What we found is that Acacia dealbata was highly distributed in the whole study area during the study period, and it was fluctuating from year to year. In the classification, we reached an overall classification accuracy of 66.9%. And looking at the proportion of Acacia dealbata in the study area, we found that actually the proportion was higher in the belts in the first 10 years and more or less higher in the protected areas in the second part from 2012 to 2020. However, overall, there was no significant difference between the proportion of Acacia dealbata in the protected area and the belt. Looking at the burned area, this plot shows 19 plots that represent 1 year each from 2001 to 2019, and the red color is the burned area. What we found is that the study area was highly affected by fires throughout the whole study period, in some years with more or some years with less fires. We found that protected areas were actually significantly more affected by fires than their 10-kilometer belts, which was contrary to our hypothesis. Looking at the tree cover loss in this area in the study period, we also found that the study area was highly affected by tree cover loss throughout the 19 years, also with sometimes more, sometimes with less tree cover loss. And here we found that tree cover loss was significantly higher in the 10-kilometer belts surrounding them than in the protected areas. To investigate the effect of fire and tree cover loss on the presence of Acacia dealbata, we selected 400 random plots, 200 in the protected areas and 200 in the belts. We analyzed if the number of years after a plot was affected by fire or tree cover loss had an effect on the proportion of Acacia dealbata in a plot. And here you see a plot that shows the proportion of Acacia dealbata per plot between 2010 and 2020, and the number of years on the x-axis, the number of years after a fire event. What we found was that Acacia dealbata increased after fire events and reached a maximum of proportion cover with 5 years or more than 5 years after a plot was affected by fire. On the other hand, we applied the same for tree cover loss. You see the number of years after tree cover loss on the x-axis, and we found here a different shape. We found a hump-shaped curve of Acacia dealbata after tree cover loss. We also investigated if there was a difference between if a plot was in a protected area or not, but there was no significant difference if it was protected or not. The results of the questionnaire told us that the management of implementation was only done for a short period of time and only like around 5 years back, started 5 years back. And that there was no specific budget assigned for the management of Acacia del Bata in these protected areas. Also, we found that the management has been conducted in public, but not in private property. And we also learned from the questionnaire that there is a lot of private properties in these protected areas, and thus they were just not accessible for management. To conclude this chapter, we conclude that Acacia del Bata was already present in the study area, in the protected area, and in the belt before the beginning of our study period. And further, we conclude that protected areas have largely been permeable to non-native species, to the colonization of Acacia del Bata. And last, that protected areas haven't prevented from fire and tree cover loss. With this, I come to a general discussion of the results of my thesis and discussion of some limitations of methods that I applied. For example, the comparison between protected area and surrounding belts could lead to some criticism. The sampling effort in protected areas and belts are likely not balanced. Protected areas belt zones are often sampled more because they are more accessible, and protected areas less because they are more remote. And this could lead to a sampling bias towards the belt zones. Further, the biographical region of Europe. Europe is a region with a lot of biodiversity databases available. However, it is not representative for data or biodiversity data worldwide. Worldwide biodiversity data is unequally sampled, and some of the areas are just neglected. And thus, the interpretation of the results or a generalization of the results should be reflected carefully. And finally, non-native species databases itself. Non-native species databases have often a taxonomic bias towards groups that are actually easily detectable or that people just like. Think of birds or plant species, but they are underrepresenting species that are difficult to identify, for example, insects or spiders. To give a perspective, in my thesis, I didn't look at climate change, at variables of climate change, but actually this is an urgent and very acute topic. Climate change can lead to changing growing seasons, for example, shorter and milder winters and longer growing seasons for plants, for example. And this has implications for a non-native species colonization, establishment, and spread. Thus, it would be interesting to further explore in studies, for example, which non-native species will be able to expand the range, for example, to higher elevation or to other climatic conditions, and how this would affect protected areas colonization by non-native species. And further, it would be interesting to investigate which species that has not a negative impact on the environment at the moment will have a negative impact on the environment in the future, so which will be invasive in the future. These could be species, for example, that are grown indoors or that will be able to reproduce outdoors with changing conditions, and thus to investigate the effect on the permeability of protected areas. I think that the results of my thesis have some implications for the non-native species management in protected areas, and thus I come to some final remarks. We learned that protected area surroundings are of importance for non-native species colonizations of protected areas. And thus we emphasize to expand the focus on the surroundings of protected areas when it comes to management. For example, how to do it, or an example, or examples to give, for example, by planning of areas with natural land use around the protected areas that would actually act as a buffer against non-native species colonizations, or by prevention campaigns. This could be on a municipality level. Think of citizens' engagement projects. People would be motivated to remove, for example, invasive plant species, etc. And also, third, by awareness raising with the landowners around protected areas. These could be, for example, by workshops or trainings that would make them aware about the responsibility they have for protected area colonizations by non-native species. And with this, I close my presentation. I want to thank many people, first of all, Anne Charpentier, who was my supervisor during the 3 years and 5 months, and always gave me good inputs and advices. Will Simonson from the UN Environment WCMC, Kevin Smith from IUCN, Tim Blackburn from UCL, and Harriet Allen from the University of Cambridge, and also Ana Rodriguez, who is unfortunately not here today, but who made this project possible. Pauline Roger, who was an excellent coordinator of this project, and Virginie Pons for her company in the fieldwork, Elisabeth de Mercante, who showed me around in Portugal as well, João, Marie, my 2 colleagues in my office, the whole 3rd floor of CEF, and Maria Lumieres and Carmen Sorria from Inspire for Nature, my family, and also my partner Imanol for his emotional support, and many, many more. Thank you.
Before starting the question, we're going to move the committee there to make something more convenient for everyone. We just have our back.
Is it possible to have a 3-minute break for?
Oui, faire attention, je pense qu'on a des câbles. En as-tu un? Vous voulez qu'on recule d'eux? Vous voulez qu'on recule d'eux?
Finalement, tu vas nous voir. Oui, en fait, on a besoin de se
Mais mets-toi là, tu seras moins impressionné.
Oui, mais moi, je suis...
Non, mais elle te connaît.
Avant, c'est de faire l'écran.
L'examinateur.
C'est pour la défendre.
C'est ça, quoi.
Si vous voulez qu'on vous voit, il va vraiment falloir qu'on se calme.
On va être obligé. Je suis toujours invisible? J'arrive. Ah, là, il y a de chance. On va sentir oppressé.
Il y en a qui peut s'asseoir sur le bureau là, un là.
C'est censé la rassurer.
Ça, j'ai jamais vu ça.
Non plus.
J'avais proposé à Catherine, elle n'a pas voulu bouger la table. De bouger
la table.
Ah ouais, je disais, attends, elle va être. T'imagines si tout se débranche.
Elle avait peur, c'est ça. Elle avait peur que tout se débranche.
Mais elle ne s'attendait pas à ce qu'on vienne ici.
Donc elle a dit, je ne peux pas toucher la table.
Non, ça ira.
Non, elle va les voir. Non, elle les voit.
Oui, elle voit l'écran. Okay, we're set now. Nathalie, it's up to you. You have around 20 minutes to ask your questions. Thank you for being here.
Okay, thank you very much, Mr. President. I thank you, Kathrin and Anna, to allow me to participate to this jury. I'm sorry not to be with you, but as you know, I was in an HDR this morning, and it would have been a pleasure to meet my colleagues I haven't seen for a long time, especially Gregory and Frank, and you, Kathrin, of course. So I found your work very interesting. It has a merit to question human practices around a problem identified as a major risk for biodiversity and functioning ecosystems. Your manuscript and your talk present important results of an original research, and I think it is a significant contribution to ecology of invasions. The manuscript was very clear, very well written, pleasant to read, and well argued. And your talk was also very pedagogical, so congratulations. You already have one publication, maybe another one is submit, I don't know.
It is in preparation. Yes.
Okay. And I saw in your manuscript that you had a paper, you wrote a paper during your master thesis. Why didn't you include it in your thesis work?
So the paper, it was actually a long process that it was with a lot of work, and it was published not so long time ago. And yes, I could have included more details from this work also to my work here, also because it goes to connectivity, for example, of habitats. However, it was more on aquatic species, like, or general aquatic species focused, and I had a broader scale of species included in my work. So okay. I didn't connect this really. Yes.
Okay. So I think that your work deals with the problem of biological invasions in a very original way, by different approaches, even social sciences approaches. So it's great. And it gives very important information to policymakers. And so it's great. Now I have many questions. Maybe the first one is in your chapter 1. I wonder how the Norwegian database was performed. Do the observers made systematic inventories, or it was more opportunistic inventories, and so maybe there are bias in the number of species you found here and there?
Yes, so as I already mentioned in the talk, so this database is actually really contributed by data from research institutes, environmental agencies, but also citizen science data. What we know is that in the recent years, the contribution of citizen data, for example, has increased a lot. And citizen science data, I think it is a part of the databases, of course, but it has increased because there are more like applications available to just collect the data. And of course, this could be like an overrepresentation of some species or some taxonomic groups that would make bias in these databases, of course, yes.
But more than some taxonomic groups, I suppose that citizens, when they want to inventory species, they go in places where there is a lot of rare species or interesting species. And so maybe they didn't found, maybe you underestimate finally the number of exotic species, for example.
In the citizen science data, or what in which data you mean? Sorry.
I don't know, but you told that people have to choose. What I understood is that people choose where they do the inventories. So, you know, when a naturalist wants to inventory a taxonomic group, he goes more in the places where there are a lot of rare species and not exotic ones.
Yeah, yeah. I didn't analyze that. What was the locations where citizen data are more contributed? But really in general, the databases are where it is more public or accessible area. There are more data available, and these are in particular in Norway, like where there are cities or streets, etc. But I didn't investigate, especially where citizen science or where citizens go to collect the data. I didn't do this analysis. No.
Okay, and I found that you considered all exotic species in the same way, but you found finally differences between animal and plant species in the number in protected or around protected areas. How do you explain the differences between animals and plants? They have different dispersal capacities, of course, but maybe, I don't know, how do you have any explanations?
Yes, so I think, for example, if you, I have an explanation. For example, plants, they cannot go away. So, for example, animals, they move individually, they can hide, and it's a short time that they are, for example, in a place, whilst plants are actually there forever if it is a tree or so the strategy or the sampling towards species that are static is probably big. That would be the plants. Otherwise, also like plant species, for example, if you go along roads, you can collect many of plant species at one go, for example. Whilst animals, I can imagine that you see one example, one animal, and then you collect it or you register it in a database. But this is just like it was your chance to see it.
Okay, okay. And when you make your analysis, you study areas that have very different surfaces between protected areas and around and in the belts. So did you correct in your analysis the differences of surfaces?
So what I corrected is actually, or what I tried to correct is the bias towards an overrepresentation or of abundance data. So that's why I worked with presence data only. I didn't correct for the different areas, but I did a filter like from that I excluded belts that are smaller than the protected area, for example, bigger. You know. I have to think that are
in fact, if you
bigger than the protected area, yes.
If you find more species in one area, it could be due to the relationship between number of species and surface.
Could be a bias, yes. Could be a bias and could go to some criticism and discussion, of course.
Okay. And in chapter 2, you choose 5-kilometer belts. Why? Why 5?
Yes, it's a good question. So I did some analysis also based on the 10-kilometer belts, and I found that there was not a big difference in the end if it comes to the non-native species that are present. It's not a big changing number. Also, to make exactly what you mentioned before, like this relationship between belts and protected areas not too large, because in Europe, for example, there are many small protected areas, for example, because of a lot of human pressure on the landscape. So we have a lot of small protected areas, and to not make these disproportionately big, I selected the smaller belt than the bigger belt.
Okay, and is there a link with the dispersal capacities of the species, for example, or?
So if this influence, or what was your question?
Because if you take a belt that is too large, in fact, nothing happens because the dispersal capacity of the species is too low. And maybe so I wonder if those 5 kilometers were chosen because it is maybe the order of the dispersal capacities of some species, but
not especially. I have chosen these because of dispersal capacities, especially also because different species might have different dispersal capacities. So I didn't actually include this in this decision. No.
Okay. In page 67 of your manuscript, you showed differences among countries on the effects of the land use, diversity, etc. I don't know. I couldn't see the number of your slides, so I don't know where it was, but it is, I don't know if you remember, but you showed at 67.
Maybe the manuscript?
Yes, 67, page 67.
57, I have.
Yes. It is on the effect of the land use diversity on the richness in the protected areas. And you found different situation between the different countries that you analyze. Yes. And so I wanted to know why there are so many differences between countries. The correlation, for example, is higher for UK and Norway.
Yes, it is higher for.
No, it's less. Yes,
Belgium and, for example, Switzerland. Than for Norway, for example. Yes. Yes. Okay. So here I assume that this has to do with like population development, for example, that there is in Belgium, for example, a high land use pressure also like around protected areas or in protected areas. Norway has a lot of protected areas that are actually remote or that they are part of forest landscapes that already exist. Whilst in Switzerland or Belgium, this might be like areas with more anthropogenic pressure, with more habitats already present.
Okay. And for the chapters on acacias, I didn't really understand the impacts of fires. I think it's not very clear for me. And And I understood that it was probably due to variation of flowering intensity of the acacias, and also their phenology. So I'm right? I well understood what you, is it clear for you? And do you know how to maybe correct your data to have more robust data to analyze?
For the classification, or for the fire, or I don't understand this connection. For the classification of acacia.
In fact, it was not clear that what was the impact of the fires, and I understood in your manuscript that it was probably due to variation of flowering intensity and phenology of the trees. That's
so
right.
The result on the classification, this could be based on differences in flower phenology. I don't understand exactly what you mean with the connection to the fires, but maybe you can clarify this again once more.
It's because you gave the dates of the fires, and
yes.
On the maps, I couldn't find very obvious that the impact was high or not, because I couldn't see really the influence of fires on the images that the pictures you showed.
Yes. Yes, so the analysis of the impact of the fires was on the plot level, basically. So I investigated this effect based on if a plot was affected by fire or not, and then the proportion of acacia that was in the plot. So maybe you see that not so clear if you look on the map itself. Yes. Yeah.
Okay. Difficult. My last question is that at the museum where I work, there is a controversy between scientists. Some of them wants to eradicate all the invasive species, and some others, they say that we better have to act against causes of invasions instead of on invasive species themselves. So I wanted to know what is your opinion.
Yeah. Wow. This is a difficult debate. But I actually, I was following this debate during these 3 years of my thesis, and I think it's really a difficult question, because actually, of course, the introduction of non-native species or invasive species are human attributed, and if you look at the level, like on a species level, these species, it's not his fault, for example. However, if we look at the community level, or the level on biodiversity loss that results from these introductions, then we want to conserve the biodiversity in the planet or in a region, and then the perspective of removing or eradication is understandable. I think with this debate in general, it is important to keep in mind, like, what species are we talking about? Are these species that are mammals that have emotional values associated, or like feelings? It's important to include this in strategies to eradicate. For example, if you want to eradicate, not to make them suffer or etc. But for plants, for example, yeah, we don't know if they have a more differentiated life, but I would strongly suggest making really decisions based on what we know about emotional sensibility for animals and plants, and make decisions to not harm anyone. Yeah.
Okay, thank you very much, Kathrin.
Yeah, thank you.
Thank you, Nathalie. Eric, it's your turn now.
Yes, okay. So, well, thank you, Kathrin, once more for this very clear and synthetic oral presentation. And before asking you some questions, I would like to try to give my very general feelings about this work. Overall, I found the manuscript very clear and very well written, including the part written in French language. And the collection of this huge data set included a very wise use of existing data, but also data collected in the field during a very difficult period, as we know. And there is yet one accepted publication, maybe two, and probably I have absolutely no concern about the publication of the other parts of this work, which is of high quality. I'd like to point out that beyond the aspects strictly related to biological conservation, the main topic of this thesis may address also fundamental issues relative to processes affecting habitat permeabilities from plant and animal species. Concerning the manuscript, in the introduction, I would have appreciated to read the hypothesis a little bit earlier in the text. Yeah. We have to wait some time to find them, and it would be clarify the work. Also, since the presentation of the definitions and concepts are very factual in the manuscript, I expected some more personal considerations about, for example, the ecological significance of biological invasions in terms of processes. I will come to this later on in my questions. Concerning the first chapter, how did you estimate the proportion of native species? And another question linked to this one is, did the use of occurrence instead of abundances introduce a bias in your analysis?
So the first question, you want to know how I estimated or how I made the difference between the invasive or non-native species?
Yes.
So yes, so the invasive species are actually a subset of the non-native species, and in this first chapter, I took the advantage that they were actually classified already as invasive species. It was the species that were severe and high impact on the environment. So I had information of the Norwegian species blacklist that classified these two groups as invasive species. And so the proportion was actually clear to make. The second question, can you repeat the second question?
Yes, when using occurrences instead of abundances, would it introduce a bias in your analysis, and how can you imagine this bias?
Yes, so if I would have included occurrence or abundance data instead of occurrence data, this would have led to much more overrepresentation towards belt zones. So what I tried to reduce is actually a bias towards belt zones and protected areas that are neglected in the data collection. This would have led to maybe some stronger effects in some results. But this is, yeah, this is what I wanted to reduce to just not have this bias in the data in the analysis. Yes.
Okay. And I didn't understood how you encompassed the effect of tourism or other leisure activities when considering the mechanisms responsible for this difference between the belt and the interior zone of protected areas.
So I didn't look in tourism, for example, specifically. I looked at accessibility, for example, of these protected areas, and this accessibility data I used that was actually an existing database that looked at the time that is needed to reach a protected area, and they included different variables such as transportation means or road streets between cities, etc. So, but I didn't look at tourism itself and how this would affect the non-native species introduction into the protected areas. But this could be actually an interesting question for further analysis with this data set, for example, especially.
Okay, thank you. In the second chapter, did you try to use more spatially explicit models like Morans or asymmetric eigenvector maps? Do you know these kind of multifactorial models?
These types I in specific don't know very well. No, but
okay.
No.
Possibly it would help you to demonstrate a directional pattern from the external zone towards the internal zone of protected areas. It was just it's just a suggestion. In the same chapter, I think that you say almost nothing about corridors, natural and artificial corridors. Do you think that it's important to include them explicitly in the analysis?
So natural corridors in this case, or in my chapter, the second chapter would be if protected area is surrounded by more natural landscapes. This could act as a proxy to corridors, for example. But I didn't include this especially because I used actually a broad land use level, which was the level 1 from Corinne Land cover, and I didn't go into more detailed land use levels. Also because if I go to more detailed land levels, it would actually mean that one specific record in one of these land levels would be very difficult. It would not allow with this data set because of my presence data only. Then you would probably go more for the occurrence data and this correlation analysis or this analysis.
Okay. Yes, I was thinking about, for example, road networks or hydrological networks that you can, for which you can calculate the density or the pattern, the hierarchy, I don't know. But I think it's important to see how much these corridors can go through the protected areas or not.
Yes,
and you mentioned the importance, the lack of barrier along the water bodies, especially rivers. I think that this is a good illustration of this problem.
Yes.
I come to the third chapter with the silver wattle. You have studied only blooming individuals because of course it's more easy to find them in the landscape from satellite analysis images. But do you think that you missed something, especially with young non-blooming individuals?
Of course, they are this could lead to a bias, or like this could lead to an underrepresentation, of course. However, the training, it's important to do on the blooming period, because it's actually the spectral footprint, the difference that allows to make this analysis. So could be that I missed something, but I think the training
in terms of occurrence detection.
Yeah, I think this could, it could be that it missed these species. However, the classification accuracy was based on the blooming period and the blooming period, and it's something that is shown in papers that it needed the blooming period to actually detect these species, but it could be.
Okay, maybe I'm wrong, but I think that in Portugal another species is very can be cultivated or invasive. This is eucalyptus, and this is also an Australian species. Which is in co-occurrence with the silver wattle. Did you have a look at this species also, or not?
For the classification in especially, no, but I read something about this species, and what I know is that these like species, eucalyptus and acacia, are actually both very competitive. And what studies showed is that in the end, like eucalyptus will win over acacia. It's more competitive. So in the first maybe year or 2 years, they are both competitive, and then acacia will go under. And this will make that it would not be detectable from satellite imageries anymore, because it's just an understory or just.
That's why I asked, because eucalyptus, in addition, is very sensitive to fire. So,
yeah.
Okay. Well, concerning your perspective or your conclusions, do you think that the non-native species could have a positive effect on ecosystems in the future, considering climate change? Or more generally, global change? And then the question is it relevant to fight invasive species outside the protected areas?
So of the first question of the relevance of non-native species in general, with climate change, I think, yes, if we think of climate change, it could also lead to like extinction of native species, flora or fauna, whatever. And actually then non-native species would contribute to a new diversity or new biodiversity in an area. It could also have a positive effect on some, for example, if I think of pollinators, if there is a plant species vanishing from an area which was actually an important contributor for pollination collection for some insects, and there is another species of plants that would replace this and would give the insects, for example, another opportunity, yes. The second question, can you repeat the second question, please?
Yes, it was. Do you think that it's relevant to fight against exotic species or invasive species outside from the protected areas?
So, if I want to rely on my research and my thesis output, I would say yes, because it's actually with the protected area, we want to protect the pristine nature, the pristine species that we have in a location. So my research clearly showed that there is a need to go beyond protected area boundaries to prevent non-native species colonizations in protected areas. So I would suggest that this could be an important future step to do. Yes.
Mm-hmm. And my last question: why didn't you study invasive species in southern France? I think that around Montpellier there is a lot of invasive species, and that's my question.
Yes, it's a good question. It could have been possible, and I initially also wanted to include some data from France in the analysis, but then I discovered that actually the resolution for a country-wide analysis, for example, of non-native species or invasive species, would have not, the databases of non-native species would not have fitted for my research question, because there are a lot of polygons or a lot of line where you can, yeah, it's just not such an accurate or comprehensive database for this analysis specifically. But I could have done a case study here in southern France, but maybe from my perspective, that I am from Switzerland, I was also interested to include data from Switzerland instead of southern France, for example.
Of course. Okay. Kathrin, at the end, I would say that you did a very good job during this thesis, and I congratulate you once more, as well for the written as for the oral presentation. And my personal opinion is that you have the main qualities to further develop a career of scientist. And that's all I can wish to you at the moment. Thank you.
Thank you very much, Eric. Gregory, may you pursue the defense?
Yeah, thank you. Thank you for the invitation to this jury, and thank you to Kathrin for this very interesting manuscript. Really, I had a lot of pleasure to read it. It read very easily, really. And thank you for putting Belgium on the scene. Again, you demonstrate that it's a small country, but always special in the pattern, so not like France. To answer Eric, so it was very interesting. So, in the same time, a lot of questions pop up during the reading of your thesis, mainly because I'm mainly a community ecologist. So sometimes I have some questions or remark. And first, I have some perhaps methodological remarks or recommendation on the analysis or questions on your choice. And I was a bit surprised by the common use of correlation you made across the thesis. Because you are testing hypotheses. For example, the hypothesis that the diversity of land use in the landscape influence the richness of non-native species. And you use correlation, and it was a bit a surprise for me.
Why was it a surprise? Or like, what is the point?
But correlation is not done to test hypotheses. Correlation, because with correlation, if you have a correlation, it can be simply that non-native species influence the diversity of land use in the landscape. So I would recommend you, I think the pattern will not change, but for publication, I will recommend you to use regression analysis. And also you mainly test linear relationships. Is it a choice? Do you have an hypothesis behind this choice? You have an ecological training in ecological science?
I have a training in yes, I have a background in ecological science.
So in general, in life science, are relationships linear?
No, not always. Of course, not always. No.
There are often threshold of the relationship. So do you see a way to test if that the relationships can be different than linear, and if you have some threshold? For example, it really struck me when I looked at the relationships between the diversity of land use in landscape and the number of non-native species. And for me, really, when I look at the point distribution, I see an exponential relationships, not a linear one.
Good question. Actually, I think I would need to go more maybe into statistical details also if it comes to the publication, for example. But in the first line, I was looking on linear correlation sheets, but of course there is a...
So it does not show the conclusion. You have a relationship, but I think you can find model that will better fit your data set.
Yes.
And it says something on the process, you see, because you can detect some threshold, for example, of landscape, land use diversity, while the curves will rise up. And I think in a management perspective, it's important.
Good point, yes.
And also, I would recommend you to be cautious when you merge different data sets that present different patterns.
Yes.
So again, because it was really a good example with this relationship between landscape diversity and number of species, as you said, Belgium is very exceptional. We have a very good relationships. But if you put these different countries together, then can you still claim that you detect a global pattern at European level?
Good question. Yeah. So another way would be like doing all the analysis on a country level. Then you would maybe miss the general pattern. But it's actually really challenging to put different data sets from different contributors or from different data bases together.
Because as Natalie said, also as you have a lot of data coming from opportunistic observation of species, citizen, the pattern can be different from one country to the other with different interests. So, but it's really interesting, and it was in your discussion. But probably you should insist on this. I would like to know more about the protected areas in your database because it's a bit. I would like to know if you really look what are those protected areas behind the fact they are protected. And I have very basic questions. Do you see, for example, the difference between nature preserve and Natura 2000 protected area?
Natura 2000 protected areas, they are often like, if you look at for example, at the polygons or something, they are often also big protected areas that are not for example, IUCN 1 category 1 protected areas that are small and are very like strict protected.
Exactly.
And then what is very interesting, if you see it on a map, like where is it located, for example, a Natura 2000 protected area, you would probably not expect in the remotest part of Norway, because there is like stricter protection that is possible and necessary also.
I think again, it does not change the pattern, the global pattern, but I think it's important because I can just look at the situation in Belgium. Natura 2000, as you said, are in highly anthropogenic landscape. So the objective is just to try to make coexist biodiversity and human activities. And so we don't expect non-native species to disappear or not coming in. But if it's a nature preserve is very different. It's a high level of protection, and action will be very targeted to eradication or prevention of native species, non-native species.
Yes, that's why I also wanted to, for example, in the first and second chapter, I really was strict with these categories with the strict protection, because it's just not like there are other categories like protection of monuments or something that doesn't make sense, or Natura 2000 protected areas, the pattern would be probably very much weaker than.
But here you included Natura 2000 in the database.
In the third chapter, yes.
In the protected area, yes, because I saw that Germany is covered of protected area in your map.
In which?
I saw that Germany. Was it Germany? Or
Germany, yes.
Germany, sorry.
Germany is not on my.
It's almost covered of protected area.
But this Germany, I don't know exactly how it looks like, because I didn't do analysis with Germany. But yes, that's why I included only strict protected areas.
And to your point of view, is the IUCN classification adapted to the situation of protected area in Europe. I have a lot of difficulties, for example, to put Natura 2000 protected area in the IUCN classification.
Yes, so what I see is that if they put it in the IUCN classification, they put it in maybe protected IUCN category 5 or 4, like that. This is protection that is more open, like broadly defined, or like, yeah, there is human interaction possible, for example. And yeah, the Natura 2000 network is actually really something that we have to distinguish from a strict protection. It's just not the purpose. But even there are regulations that say that the introduction of invasive species or non-native species has to be prevented or controlled. But on the same time, they want to have the land use and the interaction with the people, sustainable land use.
It's a bit this American way to see the world. We have classification, we apply it.
Yes, but this is human-made. We make classifications with the species and with the protected areas to.
Perhaps a point on using different taxonomic group together, because if I understood well, there was a lot of information in your thesis, so sometimes can make mistake. 75% of your species were plant species. So did you make some sensitivity analysis of your model by excluding plant species, other species, and see if the general pattern is still present?
No, I didn't do some analysis like that. No.
Should be interesting, I think, maybe because I was not aware of it at the beginning, but saying that 55%, 75%, sorry, of the species are plant species, I can expect that the general pattern is mainly due to plant. And I don't expect plant animals and fish to react on the same way to the permeability of the landscape.
Yeah. I think if I would like start again with my thesis, I would probably focus more on the subsets of taxonomic groups and go more into detail than doing a big generalization, because there is a risk that there is a bias or there is a distortion of the general pattern because of for example, overrepresentation.
Just a recommendation. I have a last questions. A very basic one, and I don't like your definition of invasive species. And I don't know why. Each time I come in France in a thesis on the invasive species, I have to say I don't like the definition. Do you have an idea why you don't like your definition of invasive species?
Maybe because you're a community ecologist.
No, it's because you include impact in the definition. So you use this definition of IUCN saying that invasive species are non-native species that have an impact. Why? The impact, and at the end of your thesis, it was interesting, you said, wait, and I agree with you, we don't have to have judgment on species, but impact is a judgment. And so how can we measure the impact of invasive species if we define invasive species like species that have impact on what? A part of my career has been devoted to invasive species. And when biological invasion popped up, invasive species were defined as non-native species that spread at a given rhythm across landscape. And we have Richardson, for example, gave some indication more than 100 meters from plant in 10 years, things like this. And I found it's a biological reality, and it's not an interpretation. And then among invasive species, there are some different impact.
Yes.
What do you think about this?
Yes, it is true. Of course, I also came with the perspective from like applied nature conservation because I was working together with IUCN and the WCMC, and I wanted also to focus on like what is the part of that contributes to policymakers in the end. Like there is an science policy interface. I wanted to include this in my thesis, but I agree with you that from this, like from a biological point of view or from a community ecology point, there is another way to define invasive species based on characteristics.
Because my fear is that with such definition, as scientists, we are often seen as ecological or green activists. Like we said, ecosystem services, and so okay, it's just a remark and
yes,
philosophical discussion and to have your point of view.
I'm happy for this comment.
It was just a bit surprising that you use this definition at the beginning, and then you have this very nice personal discussion. I really like it. It's important in a PhD thesis, and it was less evident, I think. Okay. Thank you. Thank you very much.
Thank you for the comments.
Thank you very much for this very interesting discussion. And would you?
Okay.
Maybe.
Okay, with pleasure. Okay, so I will not ask any question to Kathrin because I spent 3 years asking her questions, so I think it was enough time. But I would like to take a few minutes, if you agree, Richard, Frank, sorry, to put it this project in a bit larger context. And this context was an international project, Inspire for Nature, financed by Europe. And this project contained 15 PhD fellowships in 7 different European countries: France, Portugal, Spain, Italy, Denmark, England, Greece, something like that, and so on. And so your project was one of them, and all of them were at the interface between science and international policy for international conservation of biodiversity. And so for all the project, all the fellows must spend several months in a different country during their PhD project to visit and spend time in international organization of conservation. So for example, Kathrin, she spent 6 months in Cambridge, 3 months with IUCN, with Kevin Smith, and 3 months at WCNC with Will Simonson. And throughout all the projects, the Inspire for Nature project, all the fellows also participate to several meetings and workshops in several countries, always around this question of the interface between science and policy. So for sure, it was a really great opportunity for all the fellows, including Kathrin, to develop their professional project, to develop their professional networks too. But it has also a cost for all of them in terms of time, in terms of energy, in terms of traveling. And so at the end of this project, we understood that actually it was quite difficult for all the fellows to carry on at the same time their own PhD project and to participate to the very ambitious Inspire for Nature project. So it was, I think it was quite challenging, even if it was a rich and really nice context. I think it's a challenging context for PhD students. So this was the first context. The second context of this PhD start in spring 2020. Okay, another international context, but of different type. And for Kathrin, she was exactly in the middle of her fieldwork in Portugal when suddenly in few hours she had to decide to move quick before the first lockdown. And so she had to interrupt her fieldwork and to run to Spain to confine with her partner, Ivan. So it was another quite a big challenge. And then she entered into the long tunnel as everybody else, the long tunnel of Zoom, Skype, and video conference, and all this difficulty and stress associated with this second context. And I would like to add more recently a third context. This one is not an international one, it's a really private one. It's your, let's say, biological and physical changes, quite unexpected change that we have seen. Maybe it's not easy to see today because you sit, but that we all of them we could see. And again, it's a very nice context because it's a presage a very happy future. But again, it also brings it's a share of fatigue and of stress. So I would like to say that in the face of all these changes, all these unforeseen events, I would like to say that I'm really impressed by your capacity to keep going, to keep focusing on your PhD, not abandon, keep going, stay on the course, and go to the end of your project and finish your project. And really, I think I would like to congratulate you for all of this, because I think it was really noticed. It was really a challenging context for at least these three different types of results. So really congratulations for that. And I would like just to add a few other words about your human qualities. I have to say that it was for me really a pleasure to work with you, really a pleasure to work with you from a human point of view. You really have very good relationship skill, and I am sure that your future colleagues will be really enjoying time working with you. So thank you also for this nice time. So that's it. I have no more questions for you.
I have to join my colleagues for the congratulations for the job, really good job you made in the manuscript and here today. I have very few questions. It's not my role, the role of the referees, but your subject is so interesting that I have a few questions. I cannot leave this room without asking questions. Some of them are methodological, and the last one is for application. For the method, did you try to quantify the sampling effort of observation in the protected area you studied? I see in the first chapter that you have 350,000 data records. But what about the sampling effort within and around the area, the protected areas? Because there is a potential gap of observation, and I couldn't find the information in the manuscript.
I didn't write it so explicitly, no. But we were discussing this like among the supervisors and me, really. That is, it's first of all, it's a criticism of the methodological approach with cloud zones and protected areas.
It's not a criticism at all. It's just because at the end of your slideshow, you said that outside there are more data than inside. I can understand that for citizen data. But not for people who are professional, because they love reserves, they love protected area, they go systematically there. So do you have this kind of information to quantify the observation pressure within, because you have the number of records and they are located, so I would like to know.
For this, one would need to go to the level of like who is the data contributor, of course, and making more analysis with contributors' data. And yes, but no, I didn't do more analysis with this.
So then maybe you have this kind of data somewhere, but you couldn't analyze in this direction the data set. See what you mean?
This data is actually somewhere in the database. Yes.
Okay. Because if I had been a referee of your first paper, I would have asked you to verify the data to compare with in and outside. They didn't ask you to do that?
No.
Okay, because sampling effort is something really important you have to know, and they didn't ask.
Of course, yes.
Good reviewers.
Good reviewers.
No, I really understand this point, and after like working in this topic for 3 years, I would also point more on this. I learned a lot during these 3 years, and it's actually a very, very interesting question.
It would be very interesting to have this for application purposes. To know exactly what happens within and just around these areas. Because maybe they attract also the amateurs just at the border of the area. Maybe there is a reserve effect just at the border that we don't know. Okay. I have another question of methods. It's about porosity. If you have to conclude about what is a low or high porosity for a protected area at the end of your work, what kind of metric would you use? because you have a number of species that are, or a fraction of the total number of non-native species that are around or inside. But I would expect something more normalized per kilometer of boundaries, for instance, or per year, or we don't have the date of creation of this protected area, and we have a number at the end of the process. We don't know the length, we don't know the bordering, how long it is. So what kind of metric would you use if you have 6 months more, or 9 maybe?
Yes, what kind of measure? Maybe, I don't know, it would be also challenging, but maybe as a measure of surface area, you could go to surface area, for example, or you could also go to really specific like habitats, for example, looking at specific habitats.
But if I am a forest manager, for instance, what kind of data can I under to make something more concrete? Because a number of species, sometimes it means nothing for more people, most people who are managers or stakeholders in general. But a ratio or something more, I don't know, more concrete.
Or yes, also like when I think about that, for example, in a specific time, like on a temporal time, like when was the data collected? Like what is the permeability measure or measure like in a time, in 10 years or 20 years? It also be possible.
Because I imagine that some protected area are maybe 10 years old, others are 5. And to know if you do a good job in the field, maybe you have to know what happens in the course of your effort of conservation.
Of course.
You put all these guys all together in the same box, and maybe some of them would like to know if they work well. So for the application, I think there are many things to do with this data set and maybe some more applicated manuscripts. I agree with Eric about the corridors. And maybe you have these 4 countries to compare them, but I think that the state of the politics about corridors is not the same. And maybe you can explain the effect of the habitat mosaics around by the state of what happened with blue and green corridors in these countries. It's not synchronous. I think it's completely asynchronous. So. Did you try to make more geographical approaches of these problematics? Because we'd like to know the process, the mechanisms involved in this. What makes this difference so big?
I didn't try this in this thesis, but I agree it would be interesting for future work or for future research. I think there is even like a big project going on. I don't know if it's on European level or even bigger, that actually looks or wants to connect these blue-green landscapes basically. Because they are always also connected. You can look separately on different corridors, but then they influence each other as well. And yes, there is the potential with these data sets to do many more analysis, of course, and look at those specific questions. Yes.
And then this question is also for Anne. Is there something happening after Kathrin with this data set? Or if the story is at the end? Because there are many doors to open now for a practical point of view.
If you give me some funding, I'm sure.
Okay. Maybe a last one. Yes, about you looked at the part of the non-native species who were coming from around and went inside the protected area. And you conclude with the proportion. I would have guessed some more qualitative approach would be really interesting, because I think it's not a random subset of the species, but something more oriented with kind of characteristics of the species who are able to go through. And did you try to compare with new models, for instance, of species from the pool outside, the one who goes inside, Are they randomly from the pool, or is there a kind of orientation based on phylogenetical position, based on life history traits, based on functional groups, these kind of things? Did you try to look inside this group who are able to go inside?
Not so much in detail, no.
Know what kind of ability they have, maybe. This is interesting to know.
Yes. This goes in the direction of the question that I also got from the reviewers, that there are different these personal strategies between plants. It would be more interesting to also know more about these different strategies, or as you said, life history traits, or could be interesting, yeah, for further research. But I didn't go into these details in my work. No.
Because it's a porosity about what?
Yeah.
What kind of trait?
Yes, yes.
Okay. Thank you very much.
Thank you.
And now the jury will deliberate a few minutes. So we have to be alone in this room for a few minutes. If you can let us maybe 10 minutes, or we will go back and call you to come back to the room.
For this, I will